This meeting is about how to characterize tumors, and based on the knowledge of the tumors, we will be able to treat them correctly. So we can do that by analyzing biomarkers within the tumor piece, by characterizing, for example, the genomics, the transcriptomics, epigenomics. But also we can look for circulating biomarkers that will tell us also information about the tumor. And that you can look at circulating tumor cells, you can look at circulating tumor DNA, or even exosomes in some cases. And finally, we can also assess and functionally determine whether the biomarkers are good by using patient-derived models. These models are models that are generated from specimens of tumors that are obtained from patients that we can culture or we can inoculate into mice in order to recapitulate the tumor of a patient in a mouse and then characterize it. In terms of the cancer resistance is also linked to this concept of biomarkers. If we really understand the characteristics, the molecular characteristics of tumors, we will be able not only to know which is the correct treatment, but we will be able to predict how the tumor will escape to that treatment. So how the tumor is going to find a way to generate resistance to the treatment. We are observing, for example, now that upon several targeted treatments, there is some population of cells that are selected. There is a selective advantage for this population of cells to grow, and these cells are going to be resistant. If we know which are these cells, which are these characteristics, we will be able to prevent resistance by using another treatment or a combination of treatments. This was a session precisely on this, on resistance, in two different ways. It was two posters, one on in colon cancer and resistance to EGFR inhibitors. The other one was in breast cancer and resistance to PARP inhibitors. In both cases, it's the same thing, trying to understand the molecular mechanism of action of these compounds, try to assess how the cells are acquiring resistance, and then act accordingly to try to prevent resistance by combination of treatments. In the session about conquering resistance, what we did is try to look at different levels of resistance. It can be resistance by mutations, by genomic alterations, either by acquiring new mutations or by primary mutations that are selected, but it can be also at the level of signaling, different pathways that can circumvent the inhibition of one of the pathways due to the treatment, and in this way the tumor can escape. So these different ways of resistance is very important to understand in order to be able to treat them. So what is clear to me is that knowledge will allow us to have better treatment. So what we think is that by technological advances and also by full in-depth study of the mechanism of action of some compounds or even the biology of cancer, we will be able to have better treatments, better combination of treatments, because in this way we might be able to circumvent resistance, and also better biomarkers, maybe circulating biomarkers, that can allow us to monitor tumor evolution and tumor growth in a better way, in a way that we will be able to know exactly what we are treating at the very moment that we need to treat. I am also the representative of the EACR, European Association for Cancer Research. And I think here it's a crucial concept to try to put together ESMO and EACR to generate this translational effort where we can synergize and try to get better treatments by knowing and understanding the mechanism of action of some compounds, research, plus the clinical action towards the patient.